introduce my colleague, Manfred Auer, who is of Life Sciences Division and who is one of the premier people in the world in cryo-EM imaging.
Thank you. So that was very lovely. I learned a lot, and I say what we do sort of fits right into that. So what we do is we measure and count, and so that's going to be very important for the goal that Adam has put forward. And I guess I need to, yeah. So what I'm trying to do tonight is take you on a journey through cells. And so to take you to new horizons and basically show you how cells look like and what amazing things they are capable of. And I'm gonna have two communities that I'm gonna talk about. So I thought I liked this approach of it was the best of time, it was the worst of times, and that's been true 200 years ago, and it's probably still true. Some things never change, but it's also the best of times and worst of times in biology. It's beautiful. We have this amazing amount of data that we can now generate, and it's really daunting what we're going to do with that. Okay, so first, before I sort of run out of time, I want to thank my team. And this is really a team effort. Some people actually here in the audience from my team, and I really thank them from the bottom of my heart because they actually do the work while I'll kind of try to get money to make this all work. Okay, and also as Jay has pointed out and Adam has pointed out, this really takes a lot of collaborations, like a lot of very different people with very different skill sets, and we couldn't really just do this thing with sort of one, skill set. So I even have an artist in my lab that actually contributes to our research. So I want to tell you is why I think actually imaging matters. So if you just sort of look at this here, and you get sort of a little set you can buy yourself, it's really hard to know that this thing is about to fly. And you only know that because you can start putting the wiring diagram together, and you can start sort of appreciate what interacts with what, and that's how you sort of get to function. So I would argue that what we really need to have in biology is getting away from the old phone book and going towards sort of the Google Maps, where we really start to superimpose all this information on top of one another. And the way I tend to liken this is, if I have the topography, the highway network, all Starbucks around me, and there's a traffic jam, I can make an informed decision whether I want to take a detour or want to get a coffee. And that's really where we need to get to. If with any of those informations alone, you would not be able to make this decision. You need them all on top of one another. So one thing you have to sort of realize, I think, is that if we ought to understand life at all levels of complexities, we need to consider the very molecular, and Adam has been pointing towards the genomes and genes. We also need to understand the proteins, the gene products. We need to understand how the genes sort of work together as macromolecular machines. We need to understand how the macromolecular machines work together as a cell, and we need to understand how cells to work together in either a tissue or a community to fulfill their function. And when you do that, you really facing increasing complexity in scale. It's impossible to understand a human being at atomic level. It's too much information. You need to abstract going from level to level. And you need to sort of appreciate, or need to sort of be okay with a decreasing resolution, a decreasing level of detail, the higher you go up in the complexity. And I would argue we need lots of different imaging modalities, for example, to sort of visualize this entire process. And since you can't really beat physics and the tool set is limited, the only solution out there is to mix and match all the technologies. So that has a lot of problems going with them. But what I really wanted to start showing you is what actual cells really look like, because you have this sort of cartoon idea that cells sort of has the nucleus and the Golgi and the mitochondria and all of that. But then you actually start looking into a cell, and that's just a tiny sliver of a cell. Here's what you see. This actually is a cell that lets you hear. The rare reason why you can hear is because you have a cell like this, and it's complicated. It looks like the moon's surface. There's so many things going on here. It's really complicated. So the problem we're dealing with in biology it's incredibly complex. It's not a simple cartoon, but we'd like to understand it at cartoon level. Okay, it gets even more complicated now if you get into a tissue. And it's a little dark here, but you can still sort of see it. Now all the cells actually have different shapes, even the nuclei have different shapes. There's a lot of things happening here. So this is just about 20 cells. There is like you know, a million times more in every organ. Gets more complicated if you go into a tissue. Now it becomes incredibly difficult. Now even the tissues themselves are organized in a sort of a different, in a special way, and they all sort of need to work together. So we're really at this point now where we'd love to have a sort of a molecular understanding of something like as simple as a zebrafish embryo. We're not there yet, but what I'm trying to tell you today is we're getting there. We're sort of about to embark on those kind of endeavors. Okay. So what does it take? Well, the unsung hero always is pay attention to preserving the thing that you want to look at. There is no point at looking at something that you trashed in the sample preparation. So we spend a lot of effort trying to get a very good sample preservation. And what you can see here is you have a zebrafish embryo about 3 days old, or 5 days old here. And you can see that the different sample preparation choices that we have affect not only the level of the macromolecules, and actually this is a little protein right here that is responsible for your hearing. If that is broken, you can't hear me. It's an amazing thing. But it's also the level of organelles, the level of tissue of cells, and the level of tissues. So we need to get this step right here. There's no point to analyzing any of the samples that have sort of this kind of preservation. Okay, so what else does it take? Lots of money. These microscopes are not cheap. They're anywhere sort of in the ballpark between $1 and $6 million. And the sky's the limit. You can always add more goodies to it. And it's really hard to get that amount of money. And once you have the instrument, it's really hard to come up with a service contract of all of that. So it's really little things that's holding us back. You know, it's not the big things, it's little things holding us back. So anyway, there are 3 different microscopy approaches that I'm going to talk about. One of them is you taking one object and you're basically rotating that 140 images, rotating it from plus 70 to minus 70 degrees, and another one this way. That's called tomography, and that's very similar to a CAT scan, for those of you who are familiar with that. And what you basically do is you rotate the object and you get a 3-dimensional view, and from there on, it's all computational biology. Or you take a so-called focused ion beam approach, where you work your specimen into a block, and you literally slice off the very front end of the block, and you just look at the block first, slice it off, look at it, and so on. It's really simple, but still going to run you about $2 million to do so. Okay, and then you can do either that with an ion beam, or you can do this with a diamond knife. 2 ways of doing that. Okay. What it really takes, and that's really the crux, and here is actually where team science really is important. This is kind of the raw data right here. And this is, I've told you, this is a little protein that actually runs between what's called stereocilia, and if this protein is broken, you're deaf. Not even hard of hearing, you're deaf, you can't hear. This protein stretches and it opens up channels on one side, and that is how you can actually hear. So it's amazing that you have this little protein here that sort of really is important. But that protein is not by itself. This protein has all these other context around it that it needs to function properly. So we need to understand the whole function. We look at the protein, we look at the entire assembly. And we spend a lot of time analyzing the data. So it takes us maybe a week or 2 to get the sample preparation right. It takes us a couple of days to get the images. And then it takes us a year to analyze it. So that is really what a bottleneck is. And some of that is because we haven't quite yet tapped into the ingenuity of computer scientists. We're working on it, but it's a hard problem. Okay, so there's a number of things that we actually need to do. There's visualization. It actually turns out it's remarkably non-trivial to visualize a 3-dimensional object. You would think it should be easy, right? Just look at it. Well, think of the Amazon forest and you're trying to find one tree. How are you going to do that, right? If you just look at it, it's like many trees. They all sort of look alike. So what you have to do is you have to really sort of go in and go deep, and it's almost like a little video game. You have to sort of navigate through. And I'm going to show you a little bit of that a little later. But the most important thing is, so there are issues like data classification, data abstraction. We really get to simplified models. Once I know it's an actin filament, it's good enough that I know it's a tube. I don't have to know a lot more about it. There's annotation and there's representation. That is really where Adam's work comes in so beautifully. We need to make sense of these things. So we have voxels, we have sort of little pixels, but we don't have the knowledge. And the major part of my work is really trying to go from that little image information to understanding. And that is where the really hard work lies. Okay, so again, we can't really do this by ourselves. We need to have the supercomputing center, for example, that will allow us to now use the supercomputers to visualize those objects, and we envision that now the entire world will be able to look at these data sets, and we hope there will be some sort of a crowdsourcing. Maybe local high schools will 10 years down the line look at our data sets. That's a dream of us. Okay, I'm going to show you two examples. One is the mammary gland development in breast cancer, and then I want to show you it's a delicate dance for tissue integrity. And the question really that we're asking is, how do you go from a pre-puberty stage to a post-puberty stage in the mammary gland? And while doing so, how do you retain a functional tissue? That's a remarkably non-trivial thing to do. So you can't really study this in the tissue because it's too complicated, there's too much going on. So what we need to do is we take little pieces of the tissue, we put them in a three-dimensional culture system, and we stimulate them and watch what's happening to them. And what's happening to them is they're starting to undergo this branching morphogenesis. We start out with about 100 cells right here, and then they sort of go out to about 1,000 cells or so. And while they do so, they make this branching structure. And I want to cut this really short, but I want to tell you what we found. And this is kind of where this new imaging technology now comes in, that the very inside of this tissue is nicely preserved, and as you would expect, the lumen is filled, the mammary gland is not leaky, everything is intact. The most outer part that's undergoing branching morphogenesis, that's basically in embryonic development, is beautifully as you would expect it to be. But the inside here is very different, and it's very different from what the thing has sort of started with. And here's actually where sort of the brave new world comes in. By imaging very large areas, we're now able to look at the heterogeneity of this entire system. And we can look at the details, and for example, we found that the tissue, the cell polarity has broken down. We're getting this very unusual membrane patches. And those membrane patches are if we image this sort of in serial sectioning, it actually becomes quite extended. And this sort of becomes sort of more obvious, I think, if we look at this here, where you can see here that it's little flaps of membrane. So the cells are very loosely connected with one another, but what they do is they shake hands. So they actually has a little dance where they actually sort of shake hands. And even more interesting, there are cells in there that migrate. So within this whole thing migrating, there are cells in between that migrate. And the most fascinating part of it is you actually can see, so you can actually see the cells migrating right here. So it's going out there, it puts a little protrusion out there, and sometimes that protrusion is non-productive, but it retracts it. Say like, okay, never mind. And when it comes to the outside, it usually comes back in. But there are conditions that you can introduce where it won't come back in, where it's actually trying to go out. And here's a desperate attempt of this orange cell trying to bring the blue cell back in. There's a lot of things we don't understand, but there's a lot of things we can do now, we can observe now. And the three-dimensional imaging is really critical. We also work on breast cancer. For those of you who have been at Mina Bissel's talk, we're working with Mina on this. We get very exquisite preservation. We actually see nuclear pore complexes right here. We see individual ribosomes. It's very beautifully organized. And you have this pre-malignant, and you have a malignant system. And the cells are very close to one another, but something has happened. And that's what we like to understand. We'd like to know, how do you go from the pre-malignant to the malignant phenotype? What actually changes in the cell? That's something we really would like to understand. And in order to do so, we actually are looking at the ultrastructure, but the ultrastructure is very sensitive. However, in order to do this right, we need to do this in 3 dimensions, and we need to do lots of it. Okay, here's an example of how the cells do this little dance. These are all little hands, little fingers and hands, and it's how they actually sort of go about one another. The interesting thing is normal cells don't do that, but cancer cells do that. And we saw that embryonic cells that sort of move do the same thing. Okay, here's an example where we're trying to basically map out the cell cytoskeleton. And we got huge surprises. I don't have time to go into that, but I want to have huge surprises on the organization of those cytoskeletal elements. And we're actually entertaining the hypothesis that one of the reasons that you have cancer progression is because that cytoskeleton is altered. And this is something where basically the cell is reading out its tension. The cell actually knows it's connected into a tissue. If we now alter that transmission system, we think that the cells are thinking, well, we're no longer connected, let's move on, let's do sort of an embryonic development, and that is cancer. Okay. Okay, so the key now, I think, is to measure and count and really get an accurate account what is different between those different cells. That is one of the goals that we're working on right now. Ultimately, I think this is kind of what Adam touched upon. I think we need to observe, we need to make a model, we need to simulate, we need to predict, and we see how well our prediction matches the observation and sort of refine that model. And I think that is one of the novel things in biology. We're getting to the point now where we're starting to become more like an engineering discipline, where we're making this cycle of refinement and doing better than we sort of did before. Okay, very quickly, and this is what the New York Times luckily picked up on. We have these microbial communities, and the microbial bacteria here, they have a social life too. They're actually very social organisms. They actually don't like to hang out by themselves, because if you're by yourself, you get eaten. But if you hang out with your buddies, you're protected, you know, safety in numbers. And the biofilms are organized in very complex ways. It's not like they're growing everywhere they can. They're making very distinct architecture. Nothing more fascinating, I think, than the myxobacteria that make these very elaborate mounts and very elaborate structures. So they can be very close in a biofilm, which is one of the reasons they can't be eaten so easily. And they secrete these vesicles. And they have this form of social motility where they basically, what they're trying to do is they move around like a pack of wolves. They find their prey, they're circling around it, and then they digest it. Just as simple as that. They're very slow, but they're very persistent, and they're doing it very well. They also have adventurous motility. Some of them are able to move out by themselves. And they make these very complex fruiting bodies where hundreds of thousands of cells come together, single file, and make a mount. Very, very fascinating behavior. So we have discovered that these cells secrete out not just vesicles, but chains of vesicles. And we really were wondering what it was doing. So one of the things that we found is that in those vesicle chains, and again, this is why team science is so important, because we're able to dip into these various technologies that are out there. We found some of the proteins that are known to be transferable between bacteria are found in those structures. So they're helping each other out. They're giving each other a hand. They basically help. But they also use those to kill. They actually use this to kill other people, other species, right there. They make this network, and so we call that bacterial social networks, which is probably the reason why we got the play from the New York Times. And what you see here is that this bacteria in this biofilm have the ability to communicate with one another in a stealth mode. They kind of are able to send a message to one another, and nobody's listening in. So, you know, one of the media sort of just said, like, it's like a general using Twitter, you know, instead of a secret communication channel. Okay, I'm almost done. Here's James in my lab. He's a very talented artist. His is this depiction of what's happening here. So the E. coli cells, the guys in blue, use that to communicate with one another. They leave messages in a bottle for other species. They also leave little mines, water mines, that other species may come upon. and they use it to actively attack another species. And to sort of make that point, I want to come back to that in a minute. But another thing I want to tell you is Myxococcus actually makes this colonies in this very bizarre way. So you have these channels here of higher and lower density. When you cut through the very edge of that, you see they make themselves little compartments. And we know they use these little compartments to effectively colonize. It's like it's a highway system for them. It's the ability for them to actually go out and everybody's going the same direction. What we actually find is if you no longer have this ability to make these structures, then everybody gets in a traffic jam and nobody's going anywhere. And you get run over by your competitors. In order to study this more systematically, what we have done, and I really appreciate the internal support that LBL offers some scientists for some sort of what they consider groundbreaking research, we actually sort of used acoustic printing technology to map out a pattern of different bacteria, different strains pitched against one another. And some of them grow very close to one another, and some of them stay far apart from one another. And so we've been using, or we've been developing this, and I should say my colleague in chemical science division has developed this new mass spectrometry imaging, and we're combining that with fluorescence microscopy, we're combining with electron microscopy. Now we have 3 different eyes and ears on the same problem. And one of the things I want to show you here is this is actually the colony here. You see some cells, all these little black dots are all cells here. We're coming to the edge of that. It thins out, the very, very few cells. We're coming back to that region. And there's something in between before we sort of hitting the other colony on the other side. When you're actually sort of coming in, you see some cells, and you see this kind of pile of trash. And we really don't know what that pile of trash is, except when you do the electron microscopy, it looks like vesicles, it looks like membranes, it looks like EPS. So this guy's erecting a gigantic wall of China. Gigantic. And we don't know how to do it, you don't know why to do it, and one of the things we're trying to figure out right now with the mass spec imaging, the chemical side of this. We want to see whether there are toxins in there. We want to see whether they basically leave a trail, an unpenetrable wall that you have to chew through before you get to the good stuff. Okay? So I hope I have convinced you that imaging is a powerful discovery tool. I think we need to combine a variety of different imaging, structural imaging, chemical imaging, reporter molecule imaging. And I think by combining them, we're getting an understanding of biology like we never had before. And that's where we need computational people. We need all kinds of people. We need you. Let's see. Okay, we need the 3D Google map. My hope and dream really is we can convince Google one day they're going to do this for us. Just, and you'll be able to browse it, and you will know we're in Mixoland or we're in Bacillus subtilis land, and now we're in the interaction zone. But the computational tools is really one of the things that changes it. This is something that is really changing the game for all of us. Because now we can create terabytes of data. One data set is a terabyte of data. There is no desktop computer that can handle that. We're creating now data at the pace where you can no longer actually look at the data. And that's where the supercomputing will come in very powerful. And of course, naturally, Berkely Lab is right on top of it. Okay, thank you very much. I guess there's a famous one more. I forgot. I want to show you this movie. So this is actually Myxococcus xanthus eating E. coli. So it's Myxo. It's getting into E. coli right here, and he makes this rippling move. These guys have run out of food. They're making fruiting bodies. These guys are still under attack. These guys sort of go around, and it's just fascinating to think all the things that have to happen for these millions and millions of cells to coordinate their behavior. And if you think that soil or bacteria is boring, I hope you will actually never think the same way of soil. There is a lot happening under your feet. Thank you very much. Okay, with that, it's my pleasure to introduce Carolyn Larabelle, who is a professor at UCSF, and she's also the head of the National Center for X-ray Tomography, and she's also a member of the Physical Bioscience Division.
Thank you. Okay, I'm going to tell you about a new imaging technology that we've been developing at the Advanced Light Source, and that's LBL's synchrotron. That's the big round building that you see when you look up in